This is our second session, which is going to be spinal cord stimulation for spinal cord injury. And I'm pleased to introduce Aaron Phillips, who's our session chair. He's a good friend and has been involved in the eSTAN study. And there was a speaker 2 years ago where I said he was an up and coming scientist. And now I can say he's a famous scientist. So Aaron, thank you for being the session chair, and please introduce everybody.
Yeah, thanks so much, Tay, for the introduction to, or the invitation to be here today. So, you know, we have some of the top groups in the world represented here. All of these groups have completed or are now in the process of completing the major clinical trials in this space. So I'll give you a little summary of them. So first up will be the dual presentation from Professors Gregoire, Courtine and Jocelyne Bloch from Swiss Federal Institute of Technology and CHUV Hospital in Lausanne. The second will be the really exciting work from Professor Kristin Zhao at Mayo Clinic. And the third will be from Professor and Surgeon David Darrow at University of Minnesota. He's also co-chairing with me. All of these groups are really, you know, advancing the field in this space and helping bring this therapy towards the community and towards community deployment. I've had the opportunity to work with David, Jocelyne, and Gregoire, and I've seen the work from Kristin. I know we're going to see some great science today. You know, there's this little spin in the title about autonomic function. You know, I have to say, it's been really rewarding to be part of this space in the last 3-4 years, and it has moved forward, you know, from originally being a lot about volitional motor function to now all sorts of applications, including bladder, bowel, blood pressure control. And we know these are huge health priorities for people with spinal cord injury. David, I don't know, do you want to say anything at this point?
No, I think I agree. It's very exciting. We have super awesome players here talking about great science and the real intersection of sort of science and translation. So I think it's, you've already said it all. And you have I'm going to be a terrible co-chair.
You have an interesting role of being a co-chair and speaker. It's always very smooth when that happens. So first up is the dynamic duo, Gregoire Courtine and Jocelyne Bloch. Gregoire is a full professor at the Swiss Federal Institute of Technology. Jocelyne is a neurosurgeon and neuroscience professor based out of CHUV Hospital in Lausanne, Switzerland. They are two of the foremost neuroscientists in the world, focused on using neuromodulation approaches to improve function and health for people with neurological conditions. Their research spans the entire clinical spectrum, as you'll see, from engineering to preclinical models to clinical trials. They routinely publish in big journals such as Nature, Nature Biotech, Nature Medicine, etc. Together, Gregoire and Jocelyne co-direct the NeuroRestore platform and founded Onward, a company aimed at restoring movement, independence, and health for people with spinal cord injury. I'm hoping we hear a little bit about the new hemotherapy we developed over the past few years, for sure. So please take it away, Gregoire and Jocelyne. You guys can share your screen and go for it.
Here we go. Normally you can hear us and you can see the screen. This Zoom presentation are always a challenge, especially when you are 7 hours away from the presenter. So for us, it's almost 10 p.m., but we are very happy to be here with you because the community, especially in all the Minneapolis area, is very important to all the development we are doing. So you know that Jocelyne and I, we don't only work on spinal cord injury, but spinal cord injury is a strong focus in our development. People like David, who is one of our main participants, who had a severe contusion, very severe yet not complete. And you know that after this type of injury, when David sent a command from his brain to the region of the spinal cord to activate the muscle, the signal is interrupted. Although the main region of the spinal cord that control normally leg muscle, the lumbar region, is completely spared. So 20 years ago when I was a postdoc in Los Angeles, the idea was to just deliver electrical stimulation to reactivate this lumbar spinal cord. So you're all probably very familiar with the idea that if you apply this continuous electrical stimulation over the lumbar spinal cord of rats that should be completely paralyzed, they can walk, you turn off the stimulation, locomotion stops immediately. Back on, up, and locomotion resumes. And some people have taken the path to directly test this intervention in human. For me, it was very important to actually understand the mechanism, to have a very understood approach conceptually, but also have the best technology to optimize this stimulation for human application. So what we did is first, to understand the mechanism, use a computational model. So we built this highly realistic biophysical model of the spinal cord that allows us to understand the electrical potential induced by the stimulation. And we coupled this with this anatomically realistic model that combined all the population of afferent fibers in the dorsal roots, neurons in the spinal cord. And what we found is that the stimulation at low intensity recruit all the A alpha fibers, meaning all the large diameter afferent fibers that are connected to muscle spindle, and this before any other type of neural structures. This means that this apparently unspecific stimulation of the spinal cord, because of the biophysical property of the neural structure, is actually very specific. The stimulation, at least in silico, predicted that it would activate only large diameter afferent fibers. And because this information was so important for the next step in the translation to human, we wanted to make sure that this was real. So we developed this transgenic model that allows us to visualize the proprioceptive fibers, over the entire spinal cord, and you can see these very beautiful nerve bundles that really facilitate their depolarization with electrical stimulation. And we could do calcium imaging of the dorsal root ganglia in these transgenic animals. So here what you see is the neurons activated by the epidural electrical stimulation. And if I superimpose the fluorescent signal from proprioceptive neurons, you will see that all but one are actually proprioceptive, meaning that it seems that the stimulation almost exclusively at this intensity that are functional will activate the proprioceptive neurons. And we somehow confirm this finding in transgenic mice, because when we ablated genetically just the large diameter afferent fibers coming from the muscle, the stimulation was completely ineffective in promoting locomotion. For us, it was very important because this means that we could operate a transition from this broad stimulation that just increased excitability, that worked, but is less targeted. That is less, as we thought, efficient than the biomimetic stimulation that would recruit the dorsal root with a spatial and temporal timing that reproduce the way the brain would naturally activate the spinal cord to stand, to walk, to do many tasks. So we tested this biomimetic stimulation in rats, and that the proof of concept. Here the rat has a severe contusion. It's only 1 week after the injury, so the animal is very weak. When we stimulate continuously, it's effective, as I showed you before. The animal would step continuously, but at some point, it will collapse. And this is when we turn on this biomimetic stimulation that really reproduces the way the spinal cord is naturally activated during walking. And you can see it not only rescue walking, but also increase the vigor of the leg movement. And for us, this understanding was very important because now we were ready to operate the transition to human with a clear understanding of how to stimulate and what kind of technology were necessary to implement this treatment in people. And that's when I moved to the Swiss Federal Institute of Technology in Lausanne, because I thought that was the key area to really develop this technology, but also because in this environment was a functional neurosurgeon passionate about translation, about developing new treatment, about functional neurosurgery. And this person is next to me, also on the screen, is Jocelyne Bloch, who is the head of functional neurosurgery at the University Hospital in Lausanne. Mm-hmm. And then what happened, Jocelyne?
Yeah, that was 2012, huh?
Correct.
I remember well. So when I met Gregoire, he told me about all these fascinating projects, and his ideal was to go to human. And so I had the advantage of working in the hospital, so I could open the doors of the hospital, and we created a facility to exactly reproduce what he had done in the rats in the humans. It took a while, and also for me it was really important because he told me about this, the importance of being very precise to stimulate the different routes at the right time, at the right place. So it was important for me, being a functional neurosurgeon, to do the same in the human being. And this degree of precision was important. So we needed a modelization of the spinal cord. And first of all, we needed to really understand what was the sweet spot to stimulate the roots in order to be selective and efficient. So this model was created, that was a biophysical model and an anatomical model. And we could exactly see where the dorsal roots entered the spinal cord. And then applying currents, we could modelize really the sweet spot where the effect of stimulation was the best. And it's a bit lateral to the dorsal root entry zone, and the electrodes that we had on the market were a bit narrow, and we had difficulty to reach bilaterally these lateral parts of the dorsal root entry zones. That's why we needed to modelize a new paddle lead that was more done to execute what we were demanding it. And so this was the design of this paddle lead that is a kind of average, knowing that there is a lot of variability from one individual to the other. We had to find a compromise that was the best, but is not unfortunately the best for everyone, but is at least for some patients is the best possibility to stimulate selectively all the roots. And then, so the important thing was to implant it at the right place. And for that, again, the modelization was important, and to personalize each patient's spinal cord in order to understand where the roots were entering the spinal cord from L1 to S1 in order to optimize the placement of the spinal, the paddle array was very important. So after having implanted the paddle lead and with the electrophysiological testing during the surgical procedures, then we did it in the gate lab. And it was also very important to be able to do this space and time stimulation. And for that, we had to be a bit inventive because there were, again, no devices on the market to be able to do this kind of stimulation. So we used the technology of Medtronic, which is a bit of a complicated chain of action. It's good to work with engineers, but you see that it involves a lot, patient programmers, infrared, Bluetooth, also even electromagnetic induction. So with all this chain of devices, we finally could, with a computer, do what we wanted to do.
Yeah.
And Gregoire is going to explain what.
Yeah, which was the spatiotemporal stimulation of the spinal cord. But let's start simple. Thinking about just one muscle. Imagine you ask this participant who has a complete paralysis of the muscle on this left side to perform the knee extension. So just activate vastus lateralis muscle to perform this limb extension. He cannot do it because it's paralyzed. And what we did is develop some very special method to be able to do functional MRI of the spinal cord. So we know where are the projectome from proprioceptive neuron that innervate the vastus lateral muscle. So that's our target, the region that we want to stimulate. So we, for this, need to activate the L3 dorsal roots that project to this region. We know where is the electrode array. And what we do is create a field that will push the stimulation toward the L3 roots as selectively as possible. And when you do this, you stimulate continuously. And when the participant aims to perform the movement, he executes this full leg extension against the direction of gravity on demand. This means that the residual pathway from the brain are becoming functional. And this is really like a general concept because here we target the vastus lateral muscle, but you can target in a way any muscle in the spinal cord region that you aim to modulate with the stimulation. For example, because our lead is longer, we can target the low thoracic dorsal roots to modulate trunk muscle. This is a participant with a classification as Asia A, who is not able, who have a lot of difficulty to perform this trunk movement without stimulation. And when the stimulation is turned on, you can see that it's become much easier. This is just a continuous stimulation. You really facilitate the residual pathway and is controlling his trunk muscle. So these are like, in a way, simple tasks because are less dynamic than walking. I mean, of course, walking involves the recruitment of many different muscles with very special timing. So the challenge becomes much more difficult. And in this case, if I take you step by step through the way we design the stimulation, because we realize that people sometimes don't understand the way we stimulate. So it's nice to have this little explanation here. So here we have David, when he's intending to walk. The idea is to activate the muscle as a healthy person would activate them. So this is a pattern of muscle activity you typically observe in an able-bodied person. And because of the functional MRI, we know where these motor neurons are located in the spinal cord of each participant. So when David wants to do a whole limb flexion, we know that we need to activate hip flexor and ankle flexor. So that's these two hotspots here. So we activate these two electrode array with high frequency because flexor muscle love high frequency, and we can facilitate the whole limb flexion. Then in the next phase, you need to have weight acceptance. This is the quadriceps muscle, so located at L3, so that the electrode we target. And finally, we need to activate the triceps muscle for propulsion. So this is more at the sacral level. This is the propulsion. And that's really the symphony of electrode you need to activate very precisely on the left side and the right side to replicate the way the brain activates the spinal cord in order to walk. It looks complicated, but actually because you understand the concept, it becomes very rapid to implement this kind of stimulation in patients. Right, Jocelyne?
Yeah, and it really worked, and it's impressive here. You see a patient who is in Asia A, and 10 days after the surgery, one of the first days he's trying to walk with this kind of stimulation, he's really able to perform it well. And he's not the only one. I mean, we have already, for example, here you have the example of David, who is able, you see, to walk also quite nicely on the upper screen with the space-time stimulation. When you see at the lower screen, we try to stimulate the whole spinal cord continuously, and you see that he's able to stand, but it's much more difficult for him to walk. So we are very happy to see it in these two patients, but on other patients, and also it's very, on other patients, here we have the 9 implanted patients. 6 of them are incomplete, 3 of them are complete, and all of them have this characteristic of having the possibility to walk immediately after the implant. So it's excellent to have this because you are able to train the patient
very intensively and actively.
And so when they train, not only it's good for them, they have more muscles, they feel better, but also a very good thing is that at least they see some improvement out without stimulation. Here you see this patient who was not able to move the left leg without stimulation. After 2 months of training, he's able to do it, but after 5 months, it's even better. He's able to do a full stretch of the leg. And even better, I mean, he's able to walk without stimulation, something that he would not have been able to do before the training with us. All of these patients regained a better ability. So you see on this table that those who are more impaired have less improvement, and those who are less impaired have more improvement, but all of them, even in Asia B, and
it's 22 hours
in Switzerland, and they all regain some muscle function.
And for us, it was very surprising to see this neurological recovery without the stimulation, even with very severe injury. So we really wanted to understand the underlying mechanism. So we modeled this recovery in rats. So here you have a rat with this severe contusion of the spinal cord that really severed, 10, maybe like, less than 10% of the fiber are spared. And after this type of injury, they're completely paralyzed. And when you train them, you can see some extensive recovery, animal showing stepping and balance without stimulation, showing that there's really a neurological recovery after this type of lesion. And the underlying hypothesis was that the residual pathway are reorganized in response to the intervention. And we wanted to show this with very unbiased, clear tools. So we developed these new methods to be able to clear the entire brain and spinal cord, develop new light sheet microscopes dedicated for the spinal cord, really, that enable us to scan the entire specimen without cutting it. So here you see the pathway from the motor cortex in blue, you see the reticulospinal pathways in white, the contusion, and it's completely uncut. This is one scan, there's no reconstruction here. And what we found using this type of anatomical visualization is that this active training with the highly functional state of the spinal cord below the injury induce the remodeling of the spared pathways. You see the corticospinal tract is completely interrupted. You have the retraction of the fibers, but some reticulospinal fibers are spared. And where we stimulate, this is where you see this 5-fold increase in the density of projection, showing that we really induce the growth of new nerve connection, at least the one that are spared. So, you know, I mean, this is, when you see these people, it's very impressive. It's see someone who cannot walk and is able to like take a few steps in the lab, etc. It's inspiring, and at the same time, it's very frustrating because we really want to enable them to use this type of technology in their daily life. So we develop a technological framework to enable this translation. Still the proof of concept at this level, but what happened is that people, our 9 participants, they have watches that they can connect the stimulation, even with their voice. They have many programs that enable, you know, not only standing, walking, but even using their trunk, cycling, even swimming. And they can trigger it either with clicker, so they can switch between the program, or they have a measurement unit on the feet that trigger the stimulation automatically. And that enables people like David
on. Okay, start message sent to implant.
To turn on the stimulation and now really evolved outdoors. And this is not only David, no, we have all the participants who are using this technology outside the laboratory, including people classified with clinically complete spinal cord injury, the 3 at the bottom here. You see them doing like boxing while standing, paddling on our beautiful lake in Lausanne, or like walking on a track field. So this is, you know, still at the level of the proof of concept, but you really see how this technology may very soon have an impact in the life of these people. And remember, these are individuals with chronic spinal cord injury. Some of them 13 years after the injury, and still they see this recovery. Imagine when you start early after an injury. And we know, doing experiments in rats, that when we start early, the recovery is remarkably better than when we start in the chronic stage.
Yeah, and this is something we really want to try also in human beings. And we are currently organizing a new clinical trial with acute patients, subacute patients. And we will try to implant them as soon as possible after the injury. And that's a multicentric European study that we hope to begin at the beginning of 2022. Mm-hmm.
So, I mean, I would hate to finish this presentation without talking to you about the autonomic function, as Aaron touched in the first stage, because we all know that mobility is important, but there are also the upper limb function and the autonomic function that is affected after spinal cord injury. And this is a famous video that Aaron always showed with Dr. K in Vancouver. Aaron is actually on the right here, and when you verticalize the patient, inducing an orthostatic challenge, the blood pressure drops. And Aaron observed that when you stimulate the spinal cord, you know, you can really prevent this drop in arterial blood pressure. And at the time, Aaron was very frustrated by this bit of empirical approach. And and he wanted to understand better the mechanism and of course develop technology that would enable to have a more precise modulation of the blood pressure. So I remember like he sent me a little email saying like, G, can we work together? We need to understand what's happening here and make this better. And you know, I thought it was a fantastic idea, and we had all the concept and the tools to enable this science. And I still remember this day, I pick him up at the airport, you know, and we are planning experiments here on the laptop with Team Canada, ready to address the problem of autonomic function. And the first thing, you know, was for me, I mean, we had a clear understanding that when we stimulate the spinal cord, you must recruit large diameter afferent fiber. There's no other mechanism. So what's happening? So we started decorticating, dissecting all the mechanism. And what we found is that these neurons in the intermediate lamina that are activated by the large diameter afferent fibers actually contact the sympathetic preganglionic neurons that are connected directly to the sympathetic ganglia, which create the vasoconstriction of arteries. So it was very clear, this pathway. Then we thought with Aaron, well, then the best effect will be when you have the highest concentration of these preganglionic neurons in the spinal cord. So, you know, we use all the anatomical technique in the lab in order to visualize the neuron in the sympathetic ganglia, how many are connected to the sympathetic preganglionic neurons. And then what we found, as expected, which is logical, is that the density of sympathetic preganglionic neurons is directly correlated with the blood pressure modulation in response to the stimulation of the spinal cord. I know people have been showing this before, Aaron himself, stimulating, you know, where the electrode were, meaning for the motor function at the lumbar level, and then it works. But actually the hotspots are in a very different location, in the low thoracic level. This is where we found that what we call the hemodynamic hotspot. That was the best location for the stimulation, right, Jocelyne?
Yeah, and then we tried in the monkeys.
That's correct.
With Aaron too. So this monkey had a complete transection of the spinal cord at the high dorsal level. And then I did a laminectomy to implant this paddle lead to stimulate the roots T10, T11, T12. So, and to find these hotspots with this biomimetic stimulation, we wanted to see if we also were able to challenge and go back to a normal pressure. So this monkey was had an orthostatic challenge in a vacuum chamber. And after a while, after the section, he had a drop of pressure. And you see that with the stimulation on, you can immediately have a better pressure. And when you stop the stimulation, it goes down again. And it's an immediate response. And you also see that you are able not only to trigger it for a while and go down, but you can also maintain this effect for a longer time. If you look at here at this graph, the timing is accelerated, and you can keep the pressure with the stimulation many minutes, almost 20 minutes. And when you stop the stimulation, then there is again a decrease of pressure. So it was a very interesting finding, and we also had to test this in a human being.
Yeah, but remember you were super impressed when we did this in non-human primates, right? You could not believe it was working so efficiently. And you know, Aaron was so excited by this result that he decided to test this immediately in humans. So he had this one human participant in Calgary, who is himself a surgeon, actually, one of your colleagues, in a way, who was suffering from severe orthostatic hypotension. And we really stimulated at this hemodynamic hotspot. So, you know, this is a classical test you do, you know, as you probably all know, that the blood pressure, you tilt the participant up, blood pressure goes down. Our algorithm tells that we really have to increase the stimulation amplitude, but the stimulation is not turned on yet. But when we activate the stimulation to modulate the hotspot, immediately you can rescue the blood pressure. So that was a good proof of concept. And we hope now that we can really push forward this concept and really develop a treatment to manage hemodynamics after spinal cord injury. And just to rapidly wrap up this is all what is being done in our center. So Aaron mentioned we created Neurorestore with Jocelyne to really push the translation. And our philosophy that you can see in this different circle you have seen throughout the presentation is really to go all the way from in-depth mechanism in mice to translation in human and even in application that can be commercialized and widely available for everybody who can treat patients. That's what we are doing. That's why we created a startup, Onward, together also with Aaron, who is very much involved. And Onward is developing next generation stimulation platform to really enable epidural stimulation to become a treatment for people with spinal cord injury. And we hope, sure there are some neurologists or neurosurgeon listening here. We hope that we can bring this technology to you and your clinics as soon as possible. So thank you very much. We have a big team here working together to make this a reality.
Wow, so exciting. I don't know, Tay, if you were the one that put Gregoire and Jocelyne first, but I think that was a terrible decision. They should have been last after I give my talk. So all I can say is thank you so much for that. We do have one question from Mark, a question about what about the less visible benefits, but very personal to the patient, such as incontinence, body temperature control, and things like that. I think this was asked before you started talking about some of the autonomic stuff, but I think you know.
So as you also observed, David, and all of us doing epidural stimulation in human, when you start stimulating the spinal cord, many things happen, right? Even these people with chronic injury for many years, you can improve temperature, sexual function, bladder, bowel, you know, many things can improve. It's very random though. When you just stimulate, it's going to be one patient have this improvement, another patient have this improvement, etc. This is why we feel with Jocelyne that this very precise evidence-based approach, you know, is very important because there's a mechanism underlying these effects, and they are different from different functions, different stimulation parameters, locations. So we really need to build this technology that enables to really target all the neurological function that matters for people with spinal cord injury. And it's going to take more than just stimulating the spinal cord. It's going to have to be very precise.
We have another question that's come up. Did you find it challenging and or a conflict of interest mixing your academic interests with commercial interests, I guess? How did you handle it?
That's a really good question, especially for Jocelyne with a neurosurgeon.
For MDs, it's even worse. I mean, the hospital is really taking care of this. They don't really like it. But on the other side, I think it's the best way to test things quickly. And I agree with you that if it's become commercial, it's become a bit more tricky. But at this stage, we really want to achieve something that is useful. And since we don't have any, financial benefit for now, I don't feel any problems doing it. But of course, the day it becomes more commercial, probably it will become more tricky.
Yeah. The more we're expanding on the clinical trial and their scale, the more we take a step back and involve less and less involved in the clinical trials. That's when we need Kristin to help us out to show that our technology is super useful.
Well, I think that leads to my question, which is, can you talk a little bit about I know that you had great response early on after implantation. Can you talk about any therapy that you did or plan to do with the patients once they're implanted and how you think that would, affect recovery even further?
Yeah, but by this you mean the type of rehabilitation program, right? So this is clearly our strength. No, I'm really more kind of an engineer neuroscientist. Jocelyne is a neurosurgeon.
We have a lot of people.
So this is
that they were not trained in the best way probably to have the best rehab that existing on earth.
Exactly. So we cannot wait to provide the technology to people who know how to best deliver rehabilitation. This is well, we are starting now to reach out to center in the US. And you know, the Mayo Clinic is involved with external stimulation. We'll contact you for the implanted stimulation as well, and many of you, because we really want to bring this in the US as soon as possible.
Great, sounds great.
In the interest of time, I'm going to introduce our co-chair and speaker, who's going to pivot into the speaker role. So Dr. Darrow is an award-winning neurosurgeon researcher, joined the Department of Neurosurgery at University of Minnesota after completing his residency at University of Minnesota. So he earned his MD and Master's in Public Health from the University of Texas and a BS in Physics and Mathematics from Texas A&M University. As a clinician, David treats patients with pain, movement disorders, neuropsychiatric disease, and epilepsy. And thanks to support from Minnesota's Discovery Research and Innovation Economy and the Institute of Translational Neuroscience, most of Dr. Darrow's time now is devoted to research and continuing the work he was doing. So I believe we're going to be hearing about quite a few interesting things today, including the eSTAND clinical trial, which Dr. Darrow is the PI of. There's some really interesting findings coming out of that. So David, take it away.
So yes, I'm David Darrow, finally practicing neurosurgeon here at the University of Minnesota, an assistant professor, run a couple labs, the Herman Darrow lab that focuses on the other end of the central nervous system, and then some of our work with Dr. Phillips, who's been referenced a lot, and Dr. Netoff at Hennepin County in the Traumatic Brain and Spinal Cord Research Center, where we look at the bottom of the central nervous system, which, you know, I think a few of us have referred to as really, really deep brain stimulation, but also known as spinal cord stimulation or epidural spinal cord stimulation. And I do have some relevant disclosures. Apparently I can't change my slide. Let's see here. There we go. So I've gotten research funding from Abbott. That's where the stimulators come from. We've put together some patents for how we do the optimization, which I'll talk about today. And that's actually turned into a company that's trying to do optimization. And I'm a part of that. So take that into account. And obviously I'm discussing off-label use of a medical device. It's not FDA approved for spinal cord injury. So since, you know, Dr. Courtine and Dr. Bloch and everybody's given such great talks, I think I'll just build on that. And one of the things that's really interesting about spinal cord injury is that spinal cord injury is far from just one thing. You know, if you're a clinician, you know, working with spinal cord injured patients, that they're all they're very different. And when any of the sites, you know, do stimulation, what we're doing is stimulation far below the lesion, because we're trying to, in some sense or another, whether you're, you know, Gregoire and doing spatiotemporal neuromodulation, or the rest of us just, you know, content with our off-the-shelf device, doing tonic stimulation to see if we can get some benefit. We're stimulating the bottom of the cord, essentially the segments of the spinal cord that correspond to the lower extremities. And we found that this is, you know, really interesting. Like others, I've got videos that show the day after surgery for someone that's more than 6 years out, is Asia A, no motor sensory function for 6 years. We turn on the stimulator the first time, and, you know, this is sort of what we see when we ask him to move his foot. And similarly, I don't know if you'll be able to hear it, but when we ask him to kick out and hit our hand day after surgery, first time we turn it on, and we ask him to kick out, and, you know, he's eventually able to do so. And this is just tonic stimulation. This is just a square wave stimulation that we're applying. Nothing fancy like Gregoire. But, you know, this is significant, and we found now that it's more than just small movements. It actually can play a role in functional movements. And we took a different approach in how we did the study. where instead of starting out with rehabilitation, the idea was to really focus in on what can spinal cord stimulation do itself, because most of the previous work was a combination of both, as you might imagine, would be practically applied. But the challenge is how do you properly sequence these, different therapies in the right way so that you can maximize benefit? And so in our case, when we test different types of rehabilitation, we find that, for example, with cycling, where the bike pedals itself, unless you pedal hard enough to overcome it, stimulation has a big effect. And you can see in this patient, they turned the cycling around the opposite direction, in fact, while they were pedaling. And this gets translated into statistics that are interesting, because if you just do stimulation and you cycle, we really don't see much of a change. And if you just ask someone when they're not being stimulated to cycle, try as hard as you can, we don't see change. But it's the combination of two, both the intent and motivation and stimulation, that's providing a massive change in how much power can be generated while you're cycling across. I think in these patients, this is about 8 or 9 of the patients that were able to participate in this part. And I think the important thing to remember is almost all the patients in E-stand have no sensation. So, they have no proprioceptive feedback. And so everything they do essentially requires their vision to be able to provide some feedback. And we know that, patients that don't have proprioception, even if they have completely intact motor function, have an incredibly difficult time walking. And so spinal cord injury is quite variable in terms of, these, what's experienced and the challenges. One of the exciting things that we found is after many, many months of stimulation, we started to see some of these chronic changes. And this is an Asia A patient, 17 years out from injury. And one day he came into clinic and he said, I need the stimulator, I can move without it. And this is with the stimulation off. And, even 17 years out, there's still this possibility of a neuroplastic phenomenon where you can get large gross motor coarse movements, but still without any changes in sensation. And this is, really significant, because it brings about a whole new mechanism of how, potentially things like activity-based learning and rehabilitation can be paired with neuromodulation.
David, that movie didn't play continuously through, because I've seen it a few times and that one was kind of skipped.
Yeah, I think I don't going to play it again. I don't have a lot of control
Just
over.
Try it again. If it doesn't play better, then
Sure. So yeah, pretty large movements, whereas he, specifically had flaccid paralysis before this, no movements at all. So an exciting finding. And when we look over, essentially the first 7 patients in our study, about 4 of them, so more than half, we saw, increasing gains every 4 months or so. In our study is about a year. So every 4 months, we saw this group just continuously get better. And we had another cohort that, pretty much stayed the same. There was still benefit from stimulation, but there wasn't this, increase over time. And so still small numbers. We don't really know. We don't know how many people will get this. And I think it comes back to, this idea that there's a lot of variability in spinal cord injury. And so you can look at that as a barrier, or we can, look at it as, possibilities. And I think that's sort of where we're heading. Of course, as Gregoire showed, we've done the same experience with Dr. Phillips and Dr. Krasyakov, showing that, epidural stimulation at the right place can really mitigate some of this orthostatic hypotension that we see, and really improve things like cognition during these time periods. As you might imagine, without sufficient blood pressure, your brain's not getting enough oxygen or blood flow. And that plays a lot on how well you can think. And this really, we found, is translated by patients into, I get so much fatigue earlier than I used to. I really struggle throughout the day to stay awake. And that is another aspect of that, intact sympathetic activity and cerebral autoregulation it makes a huge difference that we're really interested in trying to, improve. And, we've seen also that it seems to generally, reduce spasms when you have the ability to keep stimulation on all the time. And some of that work, that's been shown since the 90s that it has an effect on spasms. But what we're really excited about is that it improves quality of life. So far in our study, we see mass gains, physical quality of life, but across the other domains, we see, smaller changes, but still in the right direction. And that gets us excited. It's what gets us out of bed in the morning to continue to do this work. A very important factor is, a lot of people, worry about, doing more surgeries in patients with spinal cord injury because of impaired blood flow and things like that. And what we found is really it's probably about the same risk profile to, spinal cord stimulation for pain. And similarly, we've also described that, and hopefully this paper will come out relatively soon, that in fact, we don't actually increase the rate of potential arrhythmias or arrhythmic type events. We actually probably reduce them with stimulation, which is really exciting because, blood pressure regulation is a much bigger deal when you have it. But as I said, spinal cord injury is a lot of things, and you can really see this if you start looking at spinal cord injuries on MRI. And this is the mid-sagittal plane from our last paper from last year that shows, essentially a cohort of patients in our study, and you can see their spinal cords are all really different. They all have, some have, big areas of myomalacia. Some have, sort of tethering. Some have, even just little wisps of spinal cords left, and some look pretty good. And this variability is extremely important because the spinal cord is dense. It has, it carries a tremendous amount of information, and that really affects the type of the pattern and phenotype of spinal cord injury that patients have. So early on, Tay and I were really figuring out how do we optimize these devices. We got all different types of spinal cord injury potentially, and we have tons and tons of outcomes that we reference today, essentially movement across all these different muscles and blood pressure and sexual function and bladder and bowel. And, how are we going to do this? This is just incomprehensible. And so we sort of, and on the device itself, there's a tremendous amount of different degrees of freedom that you can, stimulate. You've seen a lot of the paddle electrodes today, and you can quickly calculate how many different possibilities there are just from the paddle. If you have 16 electrodes, you can put it in 3 different states. So it's essentially 3 to the, 16 or 15, depending on how many contacts you have, which is a lot of possibilities. And then the parameters of the square wave are also tremendous, especially when you start talking about interleaving different, or, some of the spatiotemporal neuromodulation that Gregoire does. There's essentially infinite different settings. When you say, well, does it even matter if you optimize, if you're kind of in the right range? Well, in this patient, all we did was we just changed the frequency of stimulation and looked at leg movements. And here we kept everything else the same and just changed the frequency. And we get dramatically different responses to our cues. We say move, raise your leg, and with different frequencies, we get sort of nonlinear changes. And that's incredibly important because this is just one very straightforward parameter. There are many more. So how do you tackle this huge dimensionality and huge space and combine it with the variability of patients? And, we sort of started down this road of this kind of multi-scalar optimization. And all I'm going to talk about today is really essentially the top, which is blinded patient preference. And the idea here was that one thing I know as a practicing surgeon is that spinal cord injury patients know a ton. They know a ton about themselves. They know a ton about the literature. They know a ton about what's going on. and they're really active in that regard. And so my thought was, well, we can't figure it out, so we're going to have to see if the patients can figure it out, because it's too many factors. And so really it comes down to a very simple principle, right? Everybody's been to an optometrist or ophthalmologist, and what it sounds like is 1 or 2, 2 or 3, 3 or 4. It's the same thing. It's very simple. It's a forced binary choice. You have to give your preference to 1 or 2, and then you ask again, is it 2 or 3? And if you use this approach, mathematically, you get the most discriminability between, between settings. The trick is how you ask the question. If you do it in a randomized way, can you get statistically valid result out of it and use that to steer? Can you tell noise from signal? And so we built an app, and I say built in quotes because, we coded on the weekend, a group of us putting it together. And what we did was we essentially used this forced binary choice, and you can model a binary response with probit, and you can come up with a curve. And as long as the domain in which you're sampling over is relatively smooth, like frequency, you can start to model this. And the more data points you add, generally speaking, the better the model becomes. And so, our thoughts were, okay, well, we're going to leave it up to patients to sort of figure this out in a lot of ways, and we're going to focus on just pulse width and frequency, and then everybody kind of has to adjust the amplitude, because the amplitude adjustment has to be done with any changes in position, because the spinal cord floats around inside the dura, and you essentially have to adjust as that thing moves around. But once we can finally ask patients questions, the idea after that is, okay, well, once we make a model, how do we figure out how to make the model better? And that's where this Bayesian optimization comes in. And essentially what we say is, okay, well, now that we have this model, we're going to test the areas that we don't really understand or that change a lot or are getting pretty good, and see if we can make it better. And so the way we did this was every month participants show up. We give them essentially 15 new settings. Some of them are for autonomic function, about 8 or 9 of them are for volitional movement settings. And we say, okay, you're going to take these 8 or 9 settings, and this is the specific order that you're going to test them in, 1 a day. And at the end of the day, you're going to fill out a survey, and the survey is going to say, was today's setting better or worse for volitional movement than yesterday? And then it's going to ask about a bunch of other domains, so that we can not only try to optimize for one, but figure out the trade-offs when you're optimizing for that outcome. And I'll show you that in a little bit. So in 2 dimensions, this is sort of what the probit models look like. Here you can see the gradient on the left essentially shows the preference score. The pluses are where we've tested, and the X's are where we're going to test next. And the error function for this is on the right. And so here you can see what it looks like, sort of as time goes on, how the model changes over time based on the response from patients. And we can sort of hone in on one area that looks really good and use that data to continue to optimize. So the principle is, if we find a setting that's statistically better, essentially we move to that setting and keep going. So every month the optimization is supposed to make things better, which also makes it challenging because every month the game gets harder. We're trying to make it better and better month by month. And so at some point, you would think that you would make it as good as it's going to get. Well, when we try this in patients, we explore a huge parameter space. We try to not make assumptions about what patients would like. So what this plot shows is essentially the color gradient is where we're testing over time. And you can see the frequency and pulse width space up there and where we sampled. And this is just for a single patient. This is the amount of exploration we have done. And the nice thing is we don't really see any adverse effects from sampling all this space. And what we generally find is that a lot of times we hone in on an area that's sort of in the frequency from about 25 to 45 hertz or so. with a much more variable pulse width, and that probably has to do with the time constants that we see for the effect. So here you can see the first 7 patients that essentially made it through optimization, and you can see sort of their overlap in frequency space. They overlap quite a bit, and here you can see the domain, the axes. This is only from 20 to 100 hertz, but we sampled up to almost 1,000 hertz, and we explored that space. So this is really trimmed down, and the overlap in frequency, they all converged independently, essentially. But pulse width was much more variable, and pulse width has a pretty direct, correlation with power. So that's probably just explainable in that regard. But what's really cool about the optimization that we really like is that we collect the same forced binary choice on other types of outcomes. So while we may be optimizing for volitional movement, we also are asking about spasticity and about their ability to transfer and what their posture looks like. And so at the end of this, we get out maps in parameter space that essentially tell us optimal outcomes for these things as well, and they show us the trade-offs. So we may optimize for volitional movement in this patient, and that might be actually not as good as if we were looking for spasticity. And these sort of personalized models, we think are, sort of the top-down approach, but also quite valid and powerful in the sense that they capture, trade-offs from neuromodulation, which on the clinical side we see, frequently. Of course, we, spent a lot of time trying to validate this approach. It's quite complicated mathematically because, as I said, patients get better and better over time. But generally speaking, we can, after every month they get new settings, so it's always that learning curve at the beginning of the month, and they're all blinded and essentially randomized. And so, but over time, our models seem to do better and better, and seem to show that they're, we're able to predict essentially what a participant will say more and more. And this is one of my favorite plots that we found, where we started seeing, okay, well, some patients had really good cross-validated prediction accuracies, and some others didn't. But when we actually stratified it by how many times the patient responded, in some, we found this really nice linear correlation between, the number of actual interactions and the total cross-validated prediction accuracy. And that really got us, excited, because it meant that there was actually a goal to push for. And when we look at it statistically, we find that, yeah, the patient preference scores matter a big deal and are very significant. And hopefully this paper will be coming out soon. So in summary, our optimization is challenging because it is messy on the front end, but we can change these qualitative scores into something quantitative, model it, set up a whole Bayesian optimization off of it, and then implement it in, quasi real time to test it, and found, really interesting results from that. I think one of the strengths of this approach is it takes a qualitative preference, a very patient-centered one, and then turns it into quantitative modeling. And then we get multi-outcome acquisition. Closed loop is an amazing approach, and one that we're all shooting for, but there are drawbacks to it, and the drawbacks are that it is very focused, right? That's the point. But there are benefits to having an approach like this, where you can capture a tremendously broad amount of information with very little effort and very high efficiency, with the ability to statistically see right through any of the noise or uncertainty. And we also, with this approach, found significant improvement with continued participation, and have been moving forward with that. And it takes a massive team. I'm sure it's not even everybody, but we've had very enthusiastic support here at the University of Minnesota.
All right. Thank you so much, Dr. Darrow. That was fantastic. We have time for a couple questions, if there are any. In the meantime, are you seeing, David, convergence between patients of certain parameters that work for certain functions, or yeah?
I would say we probably are not at the point of discriminating across functions yet. Mostly that's because we're still hiring people to help us with data analysis in mass, because as you can imagine, we collect. I didn't talk about it, but one of the things that we do is we collect a lot of data at home, and we have just tons and tons and tons of data that we still haven't even started tapping into. We're mostly, focused on the main parts of the trial, but we don't know yet. We do see, what we do see we definitely see reproducible differences between outcomes within each patient. So a pretty nice error, uncertainty, and that do show differences. And I think, the point there is, even though we started out with a huge parameter space and get relatively close between patients, one of the things that I think is important is the interaction with participants in the study makes it so that they're steering their therapy quite directly. It's a little bit asynchronous the way we do it in the trial, but in the future, that they could be able to tweak their settings. You could get close with you could essentially, the thought is you could move towards a closed loop solution, but even closed loop solutions have hyperparameters, and so you can sort of tweak these parameters with patient preference in mind, and do it in a quantitative way that uses a truly quantitative cost function. So that makes it very exciting. No matter which approach you take, you can always take participant and patients into account.
Any questions from the audience? Tay, do you want to stop, or I can ask one more?
So let me just make a couple comments.
Okay.
So I think we can conclude this session, and I want to invite everybody to come to the movie. But before we let everybody go, I think there's nothing really constraining us from continuing talking, and we still have 13 people on the panel. Well, I guess some of them are AV people, but we still have a fairly good size panel of people who presented today. And if people want to stay and talk and ask questions to anybody on the panel, and anybody from the panel can ask questions, that would be great. So we'll just leave this room open until people disappear. And then the movie, there's a link that's been posted in the chat, and you should have received an email. There's the link and the passcode. Carolyn, would you like to say a few words about the movie and the discussion that will happen after? Because this is our best attempt at trying to create a social in some way. So the movie is to create a nice topic, and then the discussion is so we can have something to talk about and to think about.
Hi, everyone. Thank you for a wonderful session this afternoon. It's been really stimulating. No, I didn't mean to have a pun there. The movie tonight is called Picture a Scientist. It was actually featured last night, coincidentally, on PBS. So it is getting significant national attention. It features a variety of aspects of gender discrimination and harassment that women scientists typically confront. And so it is very illuminating, and it's comprehensive and in-depth. So I encourage all of you to take the opportunity to view it. And then at 6:15, we will have a discussion, open discussion with anyone who would like to come. We have found these to be very fruitful over the last 6 months that we've been participating. There will be a breakout room that will be dedicated just for students, as a safe space for students, and it will be run by some of our students. So please note that for any students that might want to participate. And students are not required to go to that room. It's certainly you're welcome at any of the breakout rooms. So I hope to see you there shortly.
Thank you, Carol. Yeah, and I think we're going to open the discussion room at 6:15, but then we'll probably start breaking people out into, because depending on when people finish the movie, there will be a little bit of a little bit of difference in the times there. So, people can get a little something to eat before the discussion as well. So we'll start the breakout rooms probably around 6:30, and we'll try to make the breakout room so that there may be 5-6 people in a room. So it's nice and cozy, and people can have nice discussions. So. Okay, well, thank everybody who presented, and it was very exciting. I really enjoyed the whole session, both sessions, and the voice of the community. And I want to open it up for anybody to ask questions of anybody on the panel, and we'll just chat a little while until it sort of comes to a natural conclusion. And I would love to hear from like Matthew Roderick, and Rob, I think has left the panel.
Well, Tay, I'll go. This is Matthew. I'll go ahead and ask the question that I posed to Gregoire, knowing it was a little, maybe it was hard to interpret. I was trying to be very precise in my posing the question. He made a comment towards the end about response to someone's question about autonomic function, the less visible functions. And he said, well, the behavior changes are all over the place. And it's kind of a wild, maybe a wild west of response, probably given the heterogeneity of population and the manifestation of the injury. And so my question was, he's spent a lot of time talking about mechanism and the importance of understanding what works for what. And that seems to me to be a very long process to understand the mechanism. And so I was curious about the tension between that and developing a company and a clinical trial that is effective and commercializable. And so I'm curious if any of you have, knowing that Gregoire is gone, if any of you have a comment on that as a sort of problem, like a pragmatic problem in terms of, okay, how do you design experiments that address that heterogeneity in the population, recognizing that there's all kinds of changes that people in the population are looking for? Hopefully that unpacks my question. I don't know.
I think I understand. I can take a brief stab at this before David goes, or Tay. I think you're kind of asking about, when do you pull the trigger, and when do you stop studying the multiplicity of outcomes and mechanisms and decide to, protect your concepts enough for the commercial entities to push it forward as is the natural process and the way that medication works in our society. If that is your question, the answer is really both are important. Like we're not the only field to have at the same time something that is therapeutic and working and is ready to be approved. But also we know that there's still lots of important new research to take place so that the next generation therapies, the more sophisticated therapies, are ready for the next generation and are developed when the time is right. So I think the answer is both are important. The current therapy with current approaches is being moved forward into indication by, I think, a few groups at this point. There's a lot of interest in it, but at the same time, we still need more mechanistic work, because what about the therapies of the future? We don't want to stop with what we have now, even though it works. So I hope I didn't answer a question that wasn't there, but that's my impression.
No, Aaron, I think you did in part. I guess part of what I was trying to get at is, how do you design, like, what's the practical pathway forward to design that in a parallel way so that you can move a product forward and at the same time continue to evolve the understanding of the mechanism and how to target and how best to optimize across a population, of people that maybe someone with an L1 wants a stimulator, but so does someone with a C5. L1 ASA C and C5 ASA A, like hugely different clinical manifestations. So how is practically, I'm guessing that you're all thinking about that, at least those that are certainly closer or involved in the commercialization process.
Yeah, David, do you want to answer what you were saying? Thanks, Matt.
Oh yeah, let me just take this easy question. So yeah, I mean, I think you know where I stand, Matthew. So, I think, there are many levels to this, and I think it's sort of a balance between, understanding the risk and understanding the benefits and making that as clear as possible to patients with spinal cord injury. I think we don't have a really great understanding as the spinal cord injury becomes more and more severe, and that really has to do with risk mitigation by how the trials are performed and how they're regulated. And so I guess in my mind, from that point of view, I think there are some probably decent cut points where we can say, here's a range of spinal cord injury that makes sense to move forward with certain devices, and here's another that we should continue to work on. And I'm very much on the clinical side in the sense that I want to see change sooner than later, because I know that a lot of these approaches will get better over time, and a lot of the technology is upgradable. So I think you're going to get various opinions about that. I think there are some of us that think that we're getting close to the point where these devices are proven to be helpful, and then there are others that want them to be even better. And so I think you're going to get a range of opinions, and it really is an opinion.
I wanted to ask
both of you.
I want to ask Dr. Pore, you are doing one of the first really closed loop based upon a physiological signal. And we're also very interested in these sort of closed loop therapies. And what are the challenges with the FDA of developing an adaptive device? Because I know they've been pretty conservative about approving devices that, the only one that I really am aware of is the one, for Medtronic, where essentially it's switching between settings based upon position, based on the accelerometer. And I hope you're still here.
Yeah, no, I'm here. Well, I'm hopeful that they'll be very responsive. I've been telling my fellows that we'll be using this clinically soon. So I think, hopefully very soon, and I think they'll be very responsive. I think more importantly will be, or equally importantly will be, understanding a lot of the other variables that I didn't show, but we have seen during these two trials that have been very exciting, but also concerning from a mechanistic standpoint. The impact of medications, which we briefly mentioned some of the earlier talks, but we're seeing that significantly impact some of the responses. Can't quite figure out all the details yet. A lot of pharmacology to be done there. And then just a sideline comment about the last question. We are, this device is hopefully going to be on the market relatively soon. It could mean that we're not continuing to look at neurophysiology recording and advancing other therapies. So for example, we're actually engaged in studies right now at UCSF to look at this closed loop type of system in an acute spinal cord injury, because in acute spinal cord injury, you don't want to overstimulate, and you want to be clear of not causing any neural injury on top of what's already there. But if you want to enhance the transmission through an injured area, it's kind of nice to know what information is being transmitted and how that electricity is impacting that. So I think equally push things commercially, as well as push forward the mechanistic and applicability parameters as well. So I think I don't see any reason why they wouldn't be responsive, very responsive. That workshop that they put in place for a variety of other closed loop systems was intended to sort of set the framework, for devices of various sorts, including, you know, respirators and other critical care devices. So I think that they are well engaged and understanding of the need to automate a lot of this. And it'll certainly be a focus on our NANS meeting to have a whole session on AI and other aspects of more advanced utilization of AI in neuromodulation.
Great. Great, thank you.
Sure.
Any other questions? Well, we have another full day or half day tomorrow, and we have a movie. And if we want to get people so that they can see the entire movie before the discussion, I think we'll have to end now. So wonderful. Thanks, everybody. Thanks to Aaron and David and Carolyn and George and Lucy for chairing the sessions, and Rob Woodluck. And I will see. I invite everybody to come to the breakfast tomorrow, the yolks and eggshells for women in science and engineering, and please come to this. And that starts tomorrow at 8. So, and then we will start again with the funding opportunities by NIH and NSF at noon tomorrow. Carolyn, did you want to say one last thing? Okay, good. Hope to see you guys in the discussion tonight. Take care.